Now let's talk about renal sodium regulation. Urinary sodium excretion increases when there is an excess of sodium in the body and decreases when there is a sodium deficit. This usually happens to individuals with healthy bodies. These homeostatic responses are so precise that total body sodium normally varies by only a few percentage points despite a wide range of sodium intakes and the occasional occurrence of large losses via the skin and gastrointestinal tract. Sodium is freely filterable from the glomerular capillaries into Bowman's space and is actively reabsorbed but not secreted. As you can see in the screen, there is an equation stating that the sodium excreted is equal to the sodium filtered subtracted by the sodium reabsorbed. The kidneys can adjust sodium excretion by changing both processes on the right side of the equation. For example, when total body sodium decreases for any reason, sodium excretion decreases below normal levels because sodium reabsorption increases. In understanding the responses controlling sodium absorption is to determine what inputs initiate them. This leads us to question what variables are receptors actually sensing. The answer is there are no important receptors capable of detecting the total amount of sodium in the body. Rather, the responses that regulate urinary sodium excretion are initiated mainly by various cardiovascular baroreceptors such as carotid sinus and by sensors in the kidneys that monitor the filtered load of sodium. Regulation of cardiovascular pressures by baroreceptors also simultaneously achieves regulation of total body sodium. The distribution of water between fluid compartments in the body depends in large part on the concentration of solute in the extracellular fluid. Sodium is the major extracellular solute, constituting, along with associated anions, approximately 90% of these solutes. Therefore, changes in total body sodium result in similar changes in extracellular volume, because extracellular volume comprises plasma volume and interstitial volume. Plasma volume is also directly related rather to total body sodium. Low total body sodium leads to low plasma volume, which leads to decrease in cardiovascular pressures. These lower pressures via baroreceptors initiate reflexes that influence the renal arterioles and tubules so as to decrease GFR and increase sodium reabsorption. These latter events decrease sodium excretion, thereby retaining sodium and therefore water in the body and preventing further decreases in plasma volume and cardiovascular pressures. Increases in total body sodium have the reverse reflex effects. So, the amount of sodium in the body determines the extracellular fluid volume, the plasma volume component of which helps determine cardiovascular pressures, which initiate the responses that control sodium excretion. Control of GFR. The main direct cause of the decreased GFR is a decreased net glomerular filtration pressure. This occurs both as a consequence of a decreased arterial pressure in the kidneys and, more importantly, as a result of reflexes acting on the renal arterioles. Conversely, an increase in GFR is usually elicited by neural and endocrine inputs when an increased total body sodium level increases plasma volume. This increased GFR contributes to the increased renal sodium loss that returns extracellular volume to normal.
So, for the long-term regulation of sodium excretion, the control of sodium reabsorption is more important than the control of GFR. And the major factor determining the rate of sodium absorption is the hormone aldosterone. So, to fully understand aldosterone is a steroid that acts more slowly because it induces change in gene expression and protein synthesis. And in the case of nephron, which is an important function of the kidney, the protein participates in the sodium transport. And also, the adrenal cortex produces aldosterone, which stimulates the sodium reabsorption by the distal convoluted tubule and cortical collecting ducts. So when the aldosterone is very low, approximately 2% of the filtered sodium, it is not reabsorbed but rather excreted or eliminated siya. And then when the plasma concentration of the aldosterone is high, essentially all the sodium reaching the distal tubule and cortical collecting ducts is reabsorbed. And normally, the plasma concentration of aldosterone and the amount of sodium excreted lie between these extremes. So I will try my best to explain the process of the system. So first is the activation of renin-angiotensin, and aldosterone causes renin to release from the JG cells of the kidney. So the first step, once it is activated by one of the mechanisms include the sympathetic activation and the carotid baroreceptors. So renin is released from the juxta glomerular or JG cells of the kidney. So the JG cells expresses, for example, the cells expresses a beta 1 adrenergic receptors that are therefore directly activated by the sympathetic nervous system to release renin. And also, the JG cells are stimulated to release renin when the macula-densa cells of the distal tubule detects a decrease of renal tubular sodium and chloride levels. So, second step is renin cleaves angiotensinogen that is released by the liver to form angiotensin 1. So, angiotensinogen is a globular protein produced and released by the liver. Renin is an enzyme that functions to cleave angiotensinogen to form angiotensin 1. So when uh, na-activate na ang renin-angiotensin of aldosterone, mag-release siya og renin. And the renin, which is an enzyme, it functions to cleave ang angiotensinogen, which is a globular protein, to form angiotensin 1. And the last step, angiotensinogen 1, is converted to angiotensin 2 by angiotensin converting enzyme or ACE, mainly found within the lungs. So angiotensin 2, it is the active component to activate good ang renin-angiotensin and aldosterone function to facilitate blood pressure regulation. So therefore, angiotensin 1 needs to be converted into angiotensin 2 and is accomplished by an enzyme fittingly named ACE or angiotensin converting enzyme. And this enzyme is mainly found within the lungs. So to summarize the chain of events in sodium depletion, it's pretty self-explanatory in the visual aid. So if there is an increase in renin secretion, directly increases plasma renin concentration. And also it increases the plasma angiotensin 1 concentration, then increases the angiotensin 2, and increases the aldosterone release and increases the plasma aldosterone concentration. So what are the mechanisms by which sodium depletion causes an increase in renin secretion? There are three distinct inputs to the juxtaglomerular cells or JG. First is the renal sympathetic nerves. Second is the intrarenal baroreceptors and the macula densa. So I will try my best to explain the concept of the pathways by which decreased plasma volume leads to the renin-angiotensin system, aldosterone to increase sodium reabsorption. The figure can be found in the ebook in page 512 for reference. So first, when the blood pressure in the kidneys decreases, as occurs when plasma volume also decreases, the cells are stretched less and therefore secrete more renin. So the glomerular or JG cells respond simultaneously to the combined effects of the sympathetic outputs that triggered by the baroreceptor external to the kidneys and then to their own pressure sensitivity. So second, the macula densa, it senses the amount of sodium in the tubular fluid flowing past it, and a decrease of sodium delivery causes a release of paracrine factors that diffuse from the macula densa to the nearby JG cells, and thereby activating them, causing to release renin. So there is an indirect way this mechanism is sensitive to change in sodium intake. So these are also factors that influence the decrease of plasma volume. First, we have if salt intake is low, there is less sodium filtered and less appear in the macula densa. And if high intake of sodium, it will cause a low rate of release of renin. And if ang blood pressure is decreased, the glomerular filtration rate is also decreased. And this will decrease the tubular flow rate such that less sodium is present to the macula densa. So this input also results in decreased renin release at the same time that the sympathetic nerves and the intrarenal baroreceptors are doing so. So by helping to regulate the sodium balance and well vice versa, also the plasma volume, the renin-angiotensin system contributes to control the atrial blood pressure. So it's very important with sodium intake and ang salt na atong gina-eat. However, it is not only the only way that which influences the arterial pressure. Just this ano lang, this discussion lang siya. So there are drugs that have been developed to manipulate the angiotensin II and aldosterone components in the system. So first is the ACE inhibitors such as the lisinopril that reduces angiotensin II production from angiotensin I by inhibiting angiotensin converting enzymes, which is ACE. Second is the angiotensin II receptor blockers such as losartan that prevents angiotensin II from binding to its receptor on target cell. So, for example, the vascular smooth muscle and the adrenal cortex. And then there are also drugs such as the eflerinone block that block the binding of aldosterone to its receptor to the kidney. And then there's also a direct renin inhibitor that have been developed that increase the production of angiotensin I. Although these classes of drugs have different mechanisms of action, they are all effective in the treatment of hypertension. So this highlights the many forms of hypertension that can contribute to the failure of the kidneys to adequately excrete sodium and water.
Atrial natriuretic peptide or ANP, sometimes referred to as atrial natriuretic factor or ANF, or atrial natriuretic hormone, is another regulator or ANH. ANP is produced and secreted by cells in the cardiac atria. Sodium reabsorption is prevented by ANP's effect on various tube segments. Additionally, it has the potential to influence the renal blood vessels to raise GFR, which further boosts the excretion of sodium. Additionally, ANP directly reduces aldosterone secretion, which raises sodium excretion. Although it makes sense that an excess of sodium in the body would enhance ANP secretion, changes in sodium concentration are not the cause of this increased secretion. Instead, utilizing the same reasoning that governs The regulation of renin and aldosterone secretion. AMP secretion rises in response to the expansion of plasma volume that comes along with a rise in body sodium. Atrial distension has increased specifically as the stimulus. Increasing sodium excretion results in an osmotic diuresis known as natriuresis. Arterial blood is a major factor influencing sodium reabsorption pressure. In a recent article, we talked about how arterial blood pressure acts as a signal for crucial reflexes that affect sodium reabsorption and involve the renin-angiotensin system and aldosterone. Now, we want to emphasize how locally the tubules are affected by artery pressure. In particular, pressure natriuresis, or an increase in sodium excretion, is a result of an inhibition of sodium reabsorption and an increase in sodium and consequently water excretion. It is unknown how exactly this direct effect is transmitted to sum it up, by inhibiting the function of the renin-angiotensin-aldosterone system and also acting locally in the tubules, elevated blood pressure reduces sodium reabsorption. Contrarily, a drop in blood pressure reduces sodium excretion by enhancing sodium reabsorption in the tubules and activating the renin-angiotensin-aldosterone pathway. It would be wise to take a moment to review this figure, which illustrates the strong causal relationship between arterial blood pressure and a blood volume. As a result, blood volume is one of the most significant long-term predictors of blood pressure. One of the key connections in these relationships is the direct impact of blood pressure on sodium excretion, as the image illustrates. According to one theory, a lot of people who acquire hypertension to do so because, for whatever reason, their kidneys are unable to discharge enough sodium in response to a normal arterial pressure. As a result, some dietary sodium retained at this normal pressure, increasing the plasma volume. As a result, the arterial pressure rises to a sufficient level to balance salt intake while also increasing the body's sodium concentration. An effective illustration of the general physiology principle that organ system activities are coordinated with one another and that controlled material exchange occurs between compartments and across cellular membranes is the integrated control of sodium balance.
The volume of water excreted is the difference between the volume reabsorbed and the volume filtered, the glomerular filtration rate. Changes in GFR brought on by baroreceptor afferent input as previously described typically have an equivalent impact on water excretion, but much like with sodium, the most important determinant in determining how much water is excreted is the rate of water absorption, and is determined by vasopressin. The vasopressin, the total body water, is primarily regulated by reflexes that change the hormone's output. A distinct group of hypothalamic neurons make vasopressin, and these neurons' axons end on the capillaries and the posterior pituitary, where they release vasopressin into the blood. Osmoreceptors and baroreceptors are the inputs to these neurons that are most significant. We now go to osmoreceptor control of vasopressin secretion. Changes in extracellular volume simultaneously elicit reflex changes in the excretion of both sodium and water. This is adaptive because the situations causing extracellular volume alterations are very often associated with loss or gain of both sodium and water in proportional amounts. In contrast, changes in total body water with no corresponding change in total body sodium are compensated for by altering water excretion without altering sodium excretion. Changes in water alone, in contrast to sodium, have relatively little effect on extracellular volume. The reason is that water, unlike sodium, distributes throughout all the body fluid compartments, with about two-thirds entering the intracellular compartment rather than simply staying in the extracellular compartment as sodium does. Therefore, on the next slide, cardiovascular pressures and baroreceptors are only slightly affected by pure water gains or losses. The major effect of water loss or gain out of proportion to sodium loss or gain is a change in the osmolarity of the body fluids. Under conditions due predominantly to water gain or loss, the sensory receptors that initiate the reflexes controlling vasopressin secretion are osmoreceptors in the hypothalamus. These receptors are responsive to changes in osmolarity. For example, you drink 2 liters of water. The excess water decreases the body fluid osmolarity, which results in an inhibition of vasopressin secretion via the hypothalamic osmoreceptors. As a result, the water permeability of the collecting ducts decreases dramatically. Water absorption of these segments is greatly reduced, and a large volume of hypoosmotic urine is excreted. In this manner, the excess water is eliminated and body fluid osmolarity is normalized. At the other end of the spectrum, when the osmolarity of the body fluids increases because of water deprivation, like a lack of water, vasopressin secretion is reflexively increased via the osmoreceptors. Water reabsorption by the collecting ducts increases also, and a very small volume of highly concentrated urine is excreted, by retaining relatively more water than solute. The kidneys help reduce the body fluid osmolarity back toward normal. Regulation of body fluid osmolarity requires separation of water excretion from sodium excretion. That it requires the kidney to secrete a urine that, relative to plasma, either contains more water than sodium and other solutes or less water than solute. This is made possible by two physiological factors: the osmoreceptors and the vasopressin-dependent water reabsorption without sodium reabsorption in the collecting ducts. In this diagram, osmoreceptor pathway that decreases vasopressin secretion and increases water excretion when excess water is ingested. The opposite events, an increase in vasopressin secretion, occurs when osmolarity increases, as during water deprivation.
Baroreceptors are a type of mechanoreceptors allowing for relaying information derived from blood pressure within the autonomic nervous system.
They sense the blood pressure and relay the information to the brain so that a proper blood pressure can be maintained. Baroreceptors are mechanoreceptors located in blood vessels near the heart that provide the brain with information pertaining to blood and pressure by detecting the level of stretch on vascular walls. As blood volume increases, vessels are stretched and the firing rate of baroreceptors increases. Vasopressin is a nonapeptide synthesized in the hypothalamus. This plays an essential role in the control of the body's osmotic balance, blood pressure regulation, and kidney functioning, as well as sodium homeostasis. A decreased extracellular fluid volume elicits an increase in aldosterone release via activation of the renin-angiotensin system. So, for example, when one has diarrhea or a hemorrhage. However, a decreased extracellular volume also triggers an increase in vasopressin secretion. This increased vasopressin increases the water permeability of the collecting ducts. More water is passively reabsorbed and less is excreted, so water is retained to help stabilize the extracellular volume. The baroreceptors transmit fewer impulses via afferent neurons and ascending pathways to the hypothalamus, and the result is increased vasopressin secretion. Conversely, increased cardiovascular pressures can cause more firing by the baroreceptors, resulting in a decrease in vasopressin secretion. With all this being said, the mechanism is an inverse relationship. As we can see here in figure 14.23, the baroreceptor pathway by which vasopressin secretion increases when plasma volume decreases. The opposite events, culminating in a decrease in vasopressin secretion, occur when plasma volume increases. So based on our previous discussion, we have learned two afferent pathways that control the vasopressin secreting hypothalamic cells. These are the osmoreceptors and from the baroreceptors. Hypothalamic cells receive synaptic inputs from many other brain areas in order for vasopressin secretion to be altered by pain, fear, or a variety of drugs. This applies to urine volumes and concentration. So, for example, when we feel nausea, this is because nausea is also a very potent stimulus of vasopressin release. Vasopressin infusions invoke nausea by similar pathways to equal degrees in motion sickness susceptible and resistant subjects. Furthermore, the central actions of vasopressin may contribute to nausea and slow wave disruption with vection. This same goes when we ingest alcoholic beverages. So alcohol is made up of ethanol, and this may cause the increase of urine volume produced following the ingestion of said alcohol. Thus, a urine volume in excess of the volume of the beverage consumed. This is why peeing is fairly common when we are out drinking too much alcohol. The vasoconstrictor effects of vasopressin acting on the blood vessels that perfuse the small intestines help to shift blood flow away from the gastrointestinal tract, thereby decreasing the absorption of ingested toxic substances.